So this is the second video, and this video is on phases of mitosis, prophase, metaphase, anaphase, telophase, and cytokinesis. Okay, so mitotic phase, M phase. Mitose means thread. Okay, and this thread-like structure is called is the spindle fiber. Okay, we will learn about how the spindle fiber is developing in the late prophase. Okay, so mitosis separates the replicated sister chromatids. Okay, it separates the two sister chromatids apart from each other. The two sister chromatids are joined together. Okay, at one point that is known as centromere. Right. See here the centromere. Okay, centromere has high repetitive DNA sequence that acts as binding site for protein. Okay, and the one spindle fiber you can say attaches here, and one spindle fiber attaches here on the kinetochore, and the kinetochore is a protein that is present on the centromere. Okay, and they two spindle fiber, they pull this chromosome apart in the opposite directions. So what happens is that the chromosome splits. Okay, the two sister chromatids, they split. Okay, so let's understand how is it taking place. Okay, so basically we have classified it into six parts: prophase, then we have classified the metaphase as prometaphase and metaphase. Okay, then anaphase, telophase, and then cytokinesis. This prophase, metaphase, anaphase, telophase, it is karyokinesis. You can remember this by this trick, that P before mat. Okay, before mating, you P. Okay, so P before mat. Okay, so prophase, metaphase, anaphase, telophase, then cytokinesis. So these all are part of karyokinesis. Okay, and then cytokinesis is there. Third number point is that the most dramatic consequence, the most dramatic outcome, okay, of a cell's entry into M phase is the condensation of the chromosome. Okay, you will learn how this condensation is changing the whole process and all. Okay, now another thing is that this mitosis, it was first observed by Strasburger in plant cell. Okay, in animal cell, Fleming observed this. Fleming is the same personality, Walther Fleming, in his book Cell Substance, Nucleus, and Cell Division. Okay, in 1878, he writes about these kinds of things, and his research laid the foundation stone for the whole cell biology, this mitosis. Okay, but there are certain things that he failed to explain. Number one is he failed to verify the sequence he deduced. Okay, secondly, he gave the word mitosyn to the spindle fibers, to the thread-like structures. Okay, and he also observed the movement of chromosomes. Okay, during mitosis. Then he resolved this issue, that how the genetic material, the two cells, okay, which are after dividing one cell, after dividing into two cells, how is it conserving its genetic material? Okay, that he resolved. Okay, but his drawback was he could not verify this sequence. Okay, that he deduced, right? So basically, now the thing is that this M phase generally takes place in the somatic cells, so it is also known as somatic division. Okay, but it also occurs in the germ cell or germinal cells, in oogenesis and spermatogenesis. Okay, in oogenesis and spermatogenesis, we saw there were three parts: growth phase, multiplication phase, and maturation phase. Right? In the multiplication phase and in the growth phase, this mitosis occurs and mitotic division occurs. Whereas in the maturation phase, meiosis occurs or meiotic division is taking place. Now it is called as equational division. This mitosis is known as equational division because the two daughter cells have the same number of chromosomes that the parent have. Okay, parent cell have. Now let's see. For example, this is the animal cell. Okay, after interphase, how we can say that this is the animal cell? Because there is centrioles. Keep it in mind, the characteristic feature of an animal cell is it will not have cell wall, it will have centrioles or centrosome or microtubule organization centers. Okay, now uh, these are the chromatin fibers you are looking at, right? Thread like structures, chromatin fibers or chromatin threads. This is the nuclear membrane. Okay, and in between the nuclear membrane there are nuclear pores. This is early prophase. Now if you see in the early prophase, the membrane of all cell organelles start disappearing. They don't disappear completely, start disappearing. At the end of the prophase, it completely disappears. The chromosomes, they take form. Okay, they take form, see, right? And the membrane, Golgi apparatus, endoplasmic reticulum, and nuclear membrane, like the membrane of Golgi apparatus, endoplasmic reticulum, and nucleus, dissolve, like they start disappearing. Okay, and after that, at the late prophase, okay, we see that asters are formed, spindle fibers are formed, and it is joined to the chromosomes. Right now, in the metaphase, you will see that this spindle fiber, for example, this chromosome, okay, is somewhat below the middle. Okay, if a metaplate, you can say, will be formed somewhere here. Okay, if here the plate will be formed in the metaphase. Okay, so this spindle fiber will contract more. This part of the spindle fiber will contract more, but the spindle fiber below this will not contract or contract less. Right? We will discuss about it in the metaphysic division. Right. So in the late prophase, you will see that the chromosomes are well developed, they are decondensed, okay, and spindle fibers are formed. Now, mm, there's a difference between spindle fiber and ester. See, if this is the centrioles, okay, the ester rays are protruding, the rays that are smaller in size and are protruding outside are known as ester rays. Okay, but the the structures that are running down throughout the cell, okay, throughout the cell, okay, from one end to the other end, they are known as spindle fiber. They are known as spindle fiber, okay. But the short ones, okay, that are protruding out are known as aster rays or micro. You can say aster microtubules, okay. So you can also say aster microtubules. This MT is known as is not mitochondria. It is microtubule, okay. So these are ester rays. Okay, there are two ester rays, two ester. Like amphibian. Amphibian means it can survive both on land and on water. In water, right? So both two. So amphi means two. Here there are two esters, right? See, one ester is right here, and one ester is right here. Okay, two esters are there. So it is known as amphiestral, right? It is known as amphiestral. Why is it known as amphiestral? Because it is an animal cell. It is an animal cell. Whereas the plant cells lack these kinds of ester. Okay, this kind of centrioles. I said the plant cells, they do not have centrioles in them. They do not have centrosomes. So the plant cells, they lack estrals. So they are known as amphiestral. Sorry, they are known as anestral. Anestral. An means absent. Okay, this means absent, and astral, as you know, astral. Right. So let's go through this once. Okay, in the prophase. Okay, SMC protein. SMC protein means, we have discussed it in the previous video, that SMC means structural maintenance of chromosome protein. So it will maintain the structure of the chromosome. How is it maintaining the structure of the chromosome? Let's see. Okay, SMC protein called as condensins. As the name suggests, condensins. So it will help in condensing, right? So bind to each sister chromatid at a multiple site to facilitate the condensation of the sister chromatid. Okay, this condensation is seen till metaphase. So for example, if this is the two sister chromatids, okay, you can see, for example, randomly there are condensins. Okay, these condensins, they pinch in. Okay, they pinch in. For example, uh, I have to condense this paper. So what will I do? Okay, from one point I will pinch it. Okay, so it has condensed, right? Condensing is, it will suck it in. Okay, so here, for example, this is the chromosome. What am I doing? I am pinching it. I am pinching it, right? So this thing, okay, this thing is carried out at different parts by the condensing. Okay, so here also condensing, here. So they suck in. Okay, and they facilitate the condensation of the two sister chromatids, right? Condensation means, जिसको हम बोलते हैं, sorry, condensation means, decondensed means फैल के रहना, और condensed means सिमट जाना, सिमटे जाना, decrease in size. For example, as I said, if I want to decrease, for example, this is the chromosome. Okay, if I want to decrease its size, if I want to shrink it, what will I do? Right? I just, for example, this is the condensin protein, and big, uh, you can say, chromosome is there. Okay, at one point, what I did with this condensin protein, I just helped it condense, right? I just helped it shrink like that. This condensin proteins help in shrinking. Okay, in this way only. So this, it facilitates the condensation of the two sister chromatids. Okay. Now, another thing is that if you see closely that the chromosomes, okay, the chromatid, okay, they replicate. Here, see, it replicated this much, okay, and here also it replicated this much, okay, and slowly and gradually it will replicate more and more and more, and then you will see that another like different chromatid has been formed. Okay, so see, the highlighted part is the half replicated chromatid. This is the half replicated chromatid. This is the half. Half, you cannot say half. You will say less replicated, right? Or in the process of replication. Or in the process of replication. After the process of replication completes, you will see that another, a different sister chromatid is formed. Right. Now we have seen the condensin, how condensin works. Okay. Now the thing is that how can we see, how can we say what is happening or what is not. Okay. First, this is fourth point. Okay, we won't jump here. First, we will jump here. Okay. First, each duplicated chromosome consists of two sister chromatids that are attached at the centromere. As we have seen, two sister chromatids that are attached at the centromere. Chromatids first appear in prophase. Okay, chromatids first they appear in the prophase, but clearly visible they are at the metaphase. Okay, they are not clearly visible yet in the prophase. Okay, but clearly visible. Yes. Now, the third thing is that the beginning. Okay, this is, we will learn about this bit later. Okay. Third, the beginning is marked by initiation of the condensation of chromosomes. Okay, it is marked by the initiation of the condensation of the chromosomes, because at first what was there? It was decondensed and it was spread out. Okay, to occupy territories in the nucleoplasm, specific territories. Okay, in the nucleoplasm. Now it starts to decondense. It starts to shrink, and when it is shrinking, we can say it is marking the beginning of the prophase. Okay, and the chromosomal material becomes untangled. It becomes untangled. Okay, if it does not become untangled, it will not properly condense or shrink, right? So the chromosomal material becomes untangled during the process of chromatin condensation. Okay, during the process of this chromatin condensation, centrosomes start to move to the opposite direction. It starts to move. Okay, it starts to move to the opposite poles, opposite direction poles. Okay, one at here, one at here. Okay, now the fourth point is the completion is marked by three things. Okay, first thing is that the chromosomal material becomes condensed fully. Okay, chromosomes are made up of two chromatids attached together at the centromere, as we have discussed here. Okay, here this is the centromere. Okay, this is the centromere. What is that? This. Okay. Now, each centrosome radiates out microtubules. What does it radiates out? Microtubules. Now, as we have seen that the smaller ones are known as astral microtubules or astral rays. Okay, and the one which are running through the cell, it is known as spindle fiber. The microtubule, astral microtubule, and the spindle fiber both combinedly are known as mitotic apparatus. What are they known as? They are known as mitotic apparatus. At the end, no Golgi body, no endoplasmic reticulum, and no nuclear core or nucleolus or nuclear membrane is visible. Okay, so you can remember this by this trick, GEM. Okay, G for uh, Golgi body, E for endoplasmic reticulum, and N for, you can say, nuclear membrane, nucleolus, and nuclear core. Now, what is not visible at all, like the endoplasmic reticulum and the Golgi body and the endoplasmic reticulum and the Golgi body, they are fragmented. They are fragmented and they are put into small vesicles. You will learn in protein trafficking how this small vesicles are helping out. Okay, they are containers. Okay, So, for example, this is a seminal vesicle. Okay, sorry, small vesicle. Okay, I have fragmented some parts and I have put it in it. Okay, so you can say that the small vesicles, okay, they contain fragmented endoplasmic reticulum and Golgi apparatus. Whereas the mitochondria and the lysosome are intact. Mitochondria, lysosome, and peroxisome. Mitochondria, lysosome, and peroxisome. Mitochondria, lysosome, and peroxisome are intact. Mitochondria, lysosome, and peroxisome are intact. What is the mitochondria, lysosome, and peroxisome are intact? Okay. So why are not they visible? Right. Why are not they visible? So the thing is that, for example, this is a bottle, right? And this is water. Okay. Now, if I put something in it, okay, you can see it. But if I start adding more and more sugar or salt or something like that, solute, the viscosity, okay, will change. The viscosity will increase, the density will increase, and you will see that inside this, okay, the parts you have put into it will become like not. It will not be visible, right? Because not because it is not present, because the viscosity is not allowing us to view that, right? So this was the whole point of mitosis, of prophase. Now we will see about metaphase, then anaphase, then telophase, then cytokinesis. Yes. Now the most important thing is Fyodor Boveri. Okay, we left it. Fyodor Boveri was the first who observed this spindle fiber radiating out. Okay, he was the first who observed this spindle fiber that is radiating out. Now, it is also that he observed this centrosomes. Okay, he observed, he said that there are two rod-like structures, okay, perpendicular to each other, that is now recognized as centrosome.